So it's a pleasure for me to introduce Dr. Taruna Madan. She is right now she is heading a unit on extramural research at ICMR New Delhi, and she also has a very active lab working on innate immunology in the National Institute of Reproductive Health in Mumbai. So let us get going on with her talk. But before that, let me just mention that she's also the president of the Indian Immunology Society. So we are very pleased to have her here.
Thank you, and a very good afternoon to you all. And Dipankar has raised your expectations, but I would like to start with, a disclaimer that today, whatever I'm talking, I have not worked on it. So, but yes, the life takes a full circle, and I started as a pharmacy graduate, like you guys, and then my destiny took me through a different route, and I'm back in Indian Council of Medical Research to support clinical trials and development of medical devices. So, and in the middle of this span, as Dipankar said, I do have a laboratory in the National Institute for Research in Reproductive and Child Health in Mumbai, which is again ICMR's premier research institute. And in that, what we were working on, I'll just give you a brief glimpse of that. What we are working on is that reproductive health, reproductive mucosa, again needs guarding through because reproductive mucosa is challenged by pathogens all the time whenever copulation or the mating act happens. So it is as similar to as your digestive tract mucosa or respiratory mucosa. So that is how the guarding has to be more because this is the place from where the new generation has to come. So the nature has devised very unique mechanism. Now, another important thing is that a mother is carrying a semi-allogenic fetus. So Dipankar mentioned about Dr. Peter Medawar. He was the one who actually proposed how a woman is able to carry a semi-allogenic fetus, in the layman's language, a foreign body, in her own body, which is not attacked by her immune system. So this is the puzzle that Peter Medawar was trying to resolve. And we took benefit of the research that has been carried out in this field, and we contributed to understand the role of innate immunity, especially the pattern recognition proteins, the humoral proteins that are guarding this region, and how they are not only contributing to the maintenance of pregnancy, successful maintenance of pregnancy, but they are also contributing to testicular immune privilege. Now, very few people know that the testicular development is taking place at puberty. So unlike the women whose reproductive tract is having all the eggs developed at the birth, and she is just releasing them one by one through her reproductive age. But with respect to men, the reproductive development happens at puberty. So the testicular antigens are again foreign antigens for the male body, and that's why testis is placed in a very unique way outside the body in a package which is immune privileged. The way our brain, our eyes are immune privileged, the same way testis is immune privileged. So to cut the story short, let's come back to the topic that we have to cover today. So I would be talking about cytokines, and I feel that they are the broad spectrum biologics. So if you have monoclonal antibodies, very specific target, but definitely some of the monoclonal antibodies that are having targets that have broad spectrum impacts will be broad spectrum biologics, but cytokines per se have multiple roles to play, and in fact the the research that is going on currently is to make them more specific for a particular use. And I will elaborate on this part by elucidating a wonderful story about IL-2, interleukin-2, which is a cytokine. So these are small proteins, 5 to 25 kilodalton. They are soluble. They can be membrane bound as well. They are here for cell-cell communication. And that is why they are called interleukins or cytokines. And I will tell you how this name has come in. Via induction of signaling through specific cytokine receptors, and I'll show you all the cytokine receptor families on the target cell surface, coupled with second messenger systems like JAK-STAT pathways. So these are kinases, and I will show you how the signaling happens. Now the signaling can happen in autocrine, paracrine, and endocrine manner. So guys, do you understand what do we mean by this kind of mechanism? Autocrine, paracrine, endocrine. Yeah, anyone? Yes.
Autocrine is the cell which is secreting it.
Yes.
Cytokine is working on itself.
Right.
And paracrine is that it is working on the neighboring cells.
Right.
And endocrine is that it is working on distant cells and transported through blood.
Excellent, excellent. Thank you so much. The term cytokine is derived from combination of two Greek words, cyto, meaning cell, and kinesis, meaning movement, and was used for the first time in 1974 by the pathologist Stanley Cohen. The large family of cytokines includes chemokines, interferons, interleukins, lymphokines, and tumor necrosis factors. I'm not going to cover details of this. I'm only going to cover the basics and tell you one example. Cytokines are produced by every cell, including embryonic cells, endothelial cells, fibroblasts, stromal cells, immune cells, like macrophages, lymphocytes, and mast cells. So every cell is secreting cytokines. Normally in picomolar concentrations that can increase up to 1,000 times. I hope you remember the cytokine storm of COVID, right? Cytokine storm of COVID-19, these values were up by 1,000 or even more than that. So which were the cytokines that were actually showing this kind of enhancement in COVID? Can anyone name them? Interferons, yes. Viruses do increase interferons, yes. But which were those which we were really bothered about? IL-6 and TNF, right? These are those cytokines that actually are also relevant for sepsis conditions, okay? And that is why the COVID-19 patients were having symptoms which were very similar to sepsis. Okay, owing to their immunomodulatory function, they, as well as their soluble receptors, are candidate biologics for prevention and therapy of various diseases. Interferon alpha and interferon type 1 was identified as early as 1957 as a protein that interfered with viral replication. So this was the first protein that was discovered. However, the term cytokine came in late 1970s. So post 1975, actually, there was a meeting which happened in the city called Interlaken in Switzerland. And they were definitely having a meeting. This was the second meeting for cytokines, and that's how they started this new labeling, deriving from the interleukin as well as the meaning interleukins. So they combined and they came out with this new system of classification of cytokines as interleukins. Interleukin 1, 2, and up to 10 was there till 1980s, and now we have many, many more. Okay, so when we classify them, we can structurally classify them into 4 types. 4 alpha helix bundle family with 3 subfamilies, which is having IL-2, interferon, and IL-10. And then you have interleukin 1 family. Then you have cysteine not cytokines or the transforming growth factor beta superfamily, and the IL-17 family. So these are 4 main families, and there are 3 subfamilies of the first 4 alpha helix bundle family, so a total of 7. Functionally, type 1, TNF alpha, interferon gamma, they are called as type 1 cytokines, and then you have type 2. You remember there is a classification for T helper cells, we call them as Th1 and Th2, and who actually came up with that classification? Do you remember? So this is Tim Mossman, who actually, he is from Rochester, NY University, and he published a JI paper where he showed that there are 2 types of T lymphocytes, Th1 and Th2, T helper lymphocytes. And from there, we get this term type 1 and type 2. And we also functionally classify the cytokines as pro-inflammatory and anti-inflammatory. So IL-1, IL-6, IL-12, interferon alpha, they are all categorized as pro-inflammatory. And TGF-beta and IL-10 are the premium anti-inflammatory cytokines. So anti-inflammatory cytokine armamentarium is very, very reduced in comparison to the inflammation. And can you tell me why? Why our body has so much of things related to inflammation and very few related to anti-inflammation? Can you think and let me know? Yeah? Any guesses? So is inflammation always bad or good?
In order to kill the particular pathogen, you need the immune cells to come at that place.
Right.
So when they come through either extravasation or chemotaxis, so that will generate heat and produces redness. So that is the basis of inflammation. So ma'am, if there is anti-inflammatory molecules in the system that are more, then it will counteract, it will not let the immune cells to act upon the infection. So
I'll show you several functions that these cytokines are contributing to the homeostasis of a living being for us as well, including humans. And that's why an inflammation is for growth. If you want to have new blood vessel development, you want pro-inflammatory cytokines, right? If you want the T lymphocytes to actually activate and proliferate and differentiate, you need these cytokines. And that is why the armamentarium of anti-inflammation cytokines is very small, but they are very potent, very potent. So I'll tell you about them in detail in a moment. So cytokines for therapy, just to give you an idea so that you understand why you need to study about cytokines. As Dipankar asked you why you need to study immunology, so why cytokines? So cytokines, as I mentioned, are useful for therapy. They can be the biologic drugs. Recombinant cytokines have been used as drugs since 2014, and the bone morphogenetic protein, BMP, is used to treat bone-related conditions. We have erythropoietin, which has been used to treat anemia. We have GCSF, granulocyte colony stimulating factor, used to treat neutropenia in cancer patients after chemotherapy. We have granulocyte macrophage colony stimulating factor, GMCSF, used to treat neutropenia and fungal infections in cancer patients. Interferon alpha, used to treat hepatitis C and multiple sclerosis. Interferon beta, used to treat multiple sclerosis. Interleukin 2, used to treat cancer. Interleukin 11, used to treat thrombocytopenia in cancer patients. And interferon gamma, used to treat CGD or chronic granulomatous disease and osteoporosis. And it seems to be a paradox that cytokines that are binding to antibodies have a stronger immune effect than the cytokine alone. And that is how this is one of the strategies when you are using cytokines for therapy, you are tagging them along with the antibodies. So cytokines in hematopoiesis. What is hematopoiesis? Generation of various blood cells, right, from the bone marrow. And here you have IL-3, GM-CSF, IL-1 and IL-6 responsible for megakaryocyte. Then you have IL-5 along with it forming eosinophil, and then you have IL-7 which is responsible for both B lymphocytes and T lymphocytes. And then you go ahead further from the granulocyte, monocyte, you get this generation of neutrophils and monocytes with various cytokines. So you have progenitors, and then you have the main immune cells coming from the hematopoiesis, and all that comes through these cytokines acting on specific cell types. The T cell differentiation. So as I mentioned, Mosman had told us about Th1 and Th2, but now we understand there are many, many types of T cells. So this is a naive T cell, and if you have IL-12 acting on it, it becomes Th1 with the T-bet as its transcription factor and secretes interferon gamma. You have IL-4 in the milieu, your naive T cell will become Th2 with the GATA3 transcription factor and will secrete further IL-4. You will have TGF-beta and IL-4, you will get a Th9 and that will secrete IL-9. The most important immunosuppressive T cells, T regulatory cell, comes when you have presence of both TGF-beta and IL-2. And it secretes IL-10 and TGF-beta. Th17, which has an important role to play in autoimmune diseases, is generated with TGF-beta and IL-6 and secretes IL-17, IL-21, and TNF-alpha. IL-6 and IL-21 generate T follicular helper cells, and IL-15 leads to natural killer T cells. So these are various T cell differentiated forms that come up because of the cytokine actions. So all these understanding is helpful in deciding which cytokine molecule to use for which kind of disease. How cytokine signaling happens? Very similar to how signaling happens in various other receptor-mediated activities. So here you have your cytokine that will bind to the receptor. So here we are showing the IL-6, which is binding to the receptor. So receptor has an extracellular domain, and then a cytoplasmic domain and a transmembrane domain. And here you can see alpha, beta, and then it is associated with a kinase where the phosphorylation happens, and then the chain starts for the signaling chain, and then you have specific gene transcription happening in the nucleus. So this is how a cytokine is able to mediate its action on in a paracrine or in an autocrine or an endocrine manner. These are cytokine receptor families. So you have immunoglobulin superfamily IL-1 receptor. You have hematopoietic growth factor type 1 family, which is IL-2 receptor. You have interferon type 2 family, interferon beta and gamma. You have tumor necrosis factors. So 7 transmembrane helix family for the CCR5, which is a chemokine. And then you have interleukin 17 receptor, IL-17R family. Another important thing to tell you about the evolution. So the chemokines are considered to be more recently evolved molecules than the cytokines. One evidence is that the chemokines that you see in humans, you may not have their homologs in the mouse. That is one evidence to tell you that they are recent. Plus, the chemokines are more responsive to a pathogen onslaught, and that's why there is a phenomena called gene duplication that has been happening for chemokines. So you will find the genes of the chemokines concentrated on certain domains of particular chromosomes, whereas cytokines would be spread out on different chromosomes. Okay. that was an interesting fact that I wanted to share with you. So this is how these type 1 cytokine receptor look like and ligands range from. So each cytokine receptor will have different affinity for these ligands, but it will serve as a ligand to carry out the signaling for all these ligands. So these are like almost like 4 and 5, 9 and almost like 15. 15 of them acting through the same receptor. And then you have type 2 cytokine receptors, again, several ligands. Then you have TNF receptor family, several ligands acting on it. IL-1 and IL-18 act through the IL-1 receptor family. And then you can see these G protein, which is chemokine ligands. Let me come to an example. So interleukin-2 is a small 15.5 kilodalton 4 alpha helical bundle cytokine produced predominantly by antigen stimulated CD4 positive T cells. What are CD4 positive T cells? Helper T cells. Wonderful. And it is also produced by CD8 positive T cells. Very nice. Natural killer cells and activated dendritic cells. IL-2 plays a critical role in the differentiation of the T helper cells into a variety of subsets, as I showed you. It promotes naive CD4 positive T cell differentiation into Th1 and Th2 cells while inhibiting the T helper 17 differentiation. And the most important thing is it can promote CD8 positive T cell and NK cell cytotoxic activity as well. This is the IL-2 receptor. And you can see here these are 3 domains, alpha, beta, and gamma. Important thing to note is that the Tregs are the most avid grabbers of interleukin-2 out of all the T cells. Okay? So these activities and affinities we have to keep in mind when we are going to use IL-2 for therapy. So IL-2 receptor is composed of 3 subunits, alpha, beta, and gamma, and we call them as CD25, CD122, and CD132. Do you remember where do you use CD25 in flow cytometry? For Tregs, for isolation of T regulatory cells. So because IL-2 receptor alpha is predominantly present on Tregs. So alpha, beta, gamma trimeric complex forms the highest affinity receptor. IL-2R alpha is unique to IL-2 and is expressed by a number of immune cells, including Tregs, including activated CD4, CD8 T cells, B cells, mature DCs, endothelial cells. The alpha chain is overexpressed compared with the beta-gamma chains. This is a standard. Unlike naive T cells, NK cells and memory phenotype CD8 cytotoxic T cells express high levels of beta and gamma. Okay, so there is a difference there. Nature has created a difference, and we will be using this difference for targeting IL-2 for tumor therapy. Okay, so Tregs constitutively express high levels of alpha chain and consume IL-2 more efficiently than effector CD4, CD8, and NK cells even at low level. And why do you think nature would have devised so? Why the Tregs have the highest affinity for IL-2 through the IL-2 receptor alpha, and the other cell types have been given a level lower than that?
Because T regulatory cell is partly to suppress the immune system, like for the energy. So that might be one of the reasons.
Absolutely, yes. So homeostasis has to be retained. So when a pathogen comes, the cytokines have to go up, the immune system has to get activated, and then the cells have to differentiate and they have to secrete antibodies or they have to secrete cytotoxic granules. But when the pathogen is gone, what are we supposed to do? We are supposed to bring back the system to homeostasis, because otherwise you will be wasting your energies unnecessarily. And also you understand, if you are continuing with your cytokine storm, what happens? You either end up in chronic inflammatory diseases, autoimmune diseases, or you have symptoms coming the way you saw in COVID-19. COVID-19, I'm sure you all are knowing and are aware that the virus was not there in your bodies after the day 4 or day 5, right? You all are aware of that? Wonderful. So what we were supposed to do in the COVID-19 patients was, you are all aware about the recovery trial? The recovery trial that took place in UK under the sponsorship of WHO. So in the recovery trial, the main drug that came out for COVID-19 was, what was the drug that was most useful for COVID-19? Come on, pharmacy guys. Huh? What? अरे हाथ खड़ा करो भाई. नहीं, वो तो वायरस के अगेंस्ट में है. What I am asking is, what was the most effective thing and the most cheapest drug available? No. Come on, guys. It was dexamethasone, the steroids. That worked wonders for all of us, right? Huh?
That was remdesivir.
Remdesivir was only for lowering the viral counts during the early stages, and even that time the clinical trials showed that it is not very effective. So it was dexamethasone that worked for you, and you were advised by the recovery trial team that you have to administer only on the day 6 of infection. And why not before? Why not before? Because dexamethasone is immune suppressive, and you are fighting with the virus. Your immune system would be compromised if you are starting your dexamethasone on day 1, right? So this was a very strict guideline given by ICMR and from all the government portals, that the dexamethasone administration has to happen from day 6 onwards, right? Okay, let's come back to our topic. So this is the timeline in understanding the biology and therapeutic application of interleukin-2. So I'll highlight, so 1976, the discovery of IL-2 as T-cell growth factor. It revolutionized the fields of basic immunology research. So do you guys know, when in vitro, we have to actually, infect the cells with HIV, we need IL-2. In absence of IL-2, your PBMCs will not get infected with HIV. Are you aware of that? Okay. So in 1980s, it was purified. Recombinant IL-2 was applied to propagate activated T cells. 1983, the cloning happened. 1985, IL-2 administration was capable of mediating tumor regression in humans after the clinical trials. 1987, the first progress report on the treatment of patients with cancer using high dose of IL-2. And then we moved on for the treatment of metastatic renal cell carcinoma, for the treatment of metastatic melanoma, and in 2000, IL-2 such as superkine or fused IL-2 and combined with other anti-cancer therapy such as GP100, vaccine, ipilimumab, etc. was being tried upon. So this was the paper landmark study, a T-cell selective interleukin-2 mutant protein, exhibits potent anti-tumor activity and is well tolerated in vivo, which showed that the targeted mutagenesis of human IL-2, a molecule was generated, BAY504798, And this had 3,000-fold in vitro selectivity for T cells over NK cells relative to wild-type IL-2. And can you tell me why will we look for downregulating the NK cell affinity, whereas upregulating the affinity towards T cells? We were using IL-2 for tumors.
There is a subset of NK cells which become CD56 bright, which are non-functional when they get a lot of IL-2.
Okay.
So CD56 bright and CD56 low is how, with respect to their ability of immune surveillance, is kind of, there's one theory there.
Sure, you are right. But here what happened was that if you are giving IL-2, even before it reaches the tumor, targeted tumor where it should be going and showing the efficacy, it was acting on the periphery. And the NK cells were triggered, activated, and you were seeing a lot of cytotoxicity. And the patients were succumbing not to cancer, but to the toxicity induced by IL-2, which was given actually for their therapy. So that was the thing, that we wanted a specific effect on the T lymphocytes, cytotoxic T lymphocytes, because they will be very specific for the tumor cells. They will secrete their cytotoxic granules only when they are seeing the specific antigen, tumor antigen, right? So pharma strategies facilitating IL-2 therapy were being tried out. So there was one therapy where IL-2 was synthesized and it was fused with Fc region to increase the IL-2 half-life. There was pegylation of IL-2 that improved half-life, but generation of consistent site-specific pegylated IL-2 was an enigma for the manufacturers, and there was a lot of batch-to-batch variation. So this trial was also shelved. Synthesized IL-2 muteins, as I mentioned, that either reduce IL-2 binding to IL-2R alpha or increase IL-2 binding to IL-2R beta on CD8 T cells. Now, why we want to reduce IL-2R alpha and we want to increase IL-2R beta? I just mentioned on my, I think, previous slide.
Alpha has consumed high level of IL-2.
Alpha is known to be highly expressed on T regulatory cells. And do you want T regulatory cells to come up in the tumor? No, right? So this is the most recent, update on IL-2 therapy. So a cytokine receptor masked IL-2 prodrug selectively activates tumor-infiltrating lymphocytes for potent anti-tumor therapy. So what is this prodrug consisting of? So it has a Fc fused with it. It has an IL-2 receptor beta there, and you have an IL-2 there. And why we want to have these two together? We are having the receptor as well as the cytokine together.
To increase its half-life and size.
We want it to go to the target to minimize the cytotoxicity. So when it is bound to its receptor already, it will not be available to show its cytotoxic effects on the periphery. Now, but if it is like that, then how are you going to achieve the anti-tumor effect? So for that, there is a cleavage site here. And you know that the tumor regions are enriched in matrix metalloproteases. And this protease, this region is a substrate for that. MMPs will act on it and will remove this IL-2 receptor beta from it. And you will have this some IL-2 Fc as the main drug. So this is prodrug and this is the main drug. And this pro-IL-2 preferentially expanded CTLs in the tumor, cytotoxic T lymphocytes in the tumor, and showed significant anti-tumor efficacy. And I'll show you the data. So here you have pro-IL-2, which is preferentially localizing to and getting cleaved effectively in tumors. So here you have serum IL-2 concentration post-treatment, and you have the prodrug and the drug alone, and you can see that you have almost similar kind of a half-life. So there is no change. And then you have a pro-IL-2 concentration, nanogram per gram tissue, and you see that it is more in the tumor site than in the other organs. And this is what we wanted to achieve. And then you have the tumor-bearing mice. They are showing increased concentrations of interferon gamma, which is a cytokine that is secreted by the cytotoxic T lymphocytes. And the non-tumor-bearing mice was not showing this kind of concentration with the prodrug. You can see the red one is your prodrug. Okay? And then you see the IL-6 concentration in the tumor-bearing mice, and you can see that the red one is the highest level in the tumor, not in the spleen or in the lung. So there is no adverse toxicity contributed by the prodrug. And then here you have non-tumor bearing mice, and you see that IL-6 concentration in the spleen and lung is many-fold lesser than if you use the drug alone, not in the prodrug form. And then you have percentage cleaved pro-IL-2, again very high in the tumor and very low in the other organs. And same way you can see it here, which is per total cleaved pro-IL-2 from all the tissues. If you normalize it, you see that the maximum 80% of your drug is actually confined to your tumor domain. And this is a real breakthrough achievement. Another example I give you, because pro-IL-2 is still at the animal level, and I wanted to give you a glimpse of how the clinical trials are being done with respect to cytokine drugs. So I give you an example. Yeah. Phase 2 randomized placebo-controlled clinical trial of interleukin-1 receptor antagonist in the intracerebral hemorrhage blocking the cytokine IL-1 in ICH. ICH is intracerebral hemorrhage, and they called it as block ICH. And here I described the whole trial to you. It was a multicentric, randomized, double-blind, placebo-controlled trial in patients with acute, spontaneous supratentorial ICH, intracerebral hemorrhage. There were 25 patients, 14 randomized to anakinra, which is your interleukin-1 receptor antagonist, 11 to placebo, and this was the mean age and the gender bias. The 100 milligram subcutaneous anakinra within 8 hours of onset was given, followed by 5 12-hourly 100 milligram subcutaneous injections. The primary outcome of the trial was edema extension distance on a 72-hour CT scan, and the secondary outcomes included plasma C-reactive protein and interleukin-6 levels. The anakinra group had higher baseline ICH volume, so it had to be adjusted for the baseline, but even after adjusting for the baseline, 72-hour edema extension distance was not significantly different in the two groups. However, we found that post hoc analysis demonstrated IL-6 was 56% lower at day 2 with anakinra, which can be an advantage for the patient. There were 10 and 2 serious adverse events in anakinra and placebo groups, so there are more with the cytokine group. So the conclusion was, we describe feasibility for delivering anakinra in acute ICH, provide preliminary safety data. However, this study lacked power to test for effects on edema, and thus further trials will be required. So guys, failures are bound to be there, although you are seeing all the effects in vitro, in vivo, and when you take it up to the trials, you are spending lots and lots of money, billions of dollars, but still the result can be like this. So there are various death valleys of an innovative medical technology, and you know all these things that I'm so sorry. Yeah, so ICMR has launched a mission, MedTech Mitra, where we handhold the innovators through all these death valleys with the help of our knowledge partners here. You can visit the ICMR website and MedTech Mitra website for more details. And I also wanted to apprise you, the government is going all out to support pharma research, and 9 pharma companies to get 5,000 crore government support in 5 years. And you see this is on the government website. Dr. Mansukh Madhya ji launched National Policy on Research and Development and Innovation in Pharma Medtech sector in India, and this is called PRIP scheme. And in this, 9 pharma companies, the commercial profit-making companies, are going to get support from government. And Dipankar has already apprised you, block your dates for 51st Immunocon that is going to happen at Indian Institute of Science from 16th to 19th October 2024, and thank you very much.
Thank you so much, Dr. Taruna. Are there any questions? So Dr. Taruna has very nicely summarized, different aspects, and then she's taken examples. 2 examples were taken. One is IL-2, and the other was Ankira, which is anti-IL-1, to give insights into about cytokines and therapy.
It reminded me of Shanferon trial that was going on with chronic myeloid leukemia.
Right.
And you have mentioned IL-2 for cancer. So any particular cancer, or apart from this chronic myeloid leukemia?
I mentioned that it was for renal cell carcinoma as well, metastatic melanoma as well. Okay. So IL-2 has been proposed for various tumor therapies, but the thing is that till date, the therapy is being marred by all these adverse effects, and that's why not being advocated for clinical applications.
Yeah,
yet.
in GCRI it was going on for quite some time, and then
Yes.
imatinib missile, it was the major
Yes.
molecule.
Yes.
So
yes.
I am not sure whether Shanferon was a recombinant molecule or just an Indian company. So Shanta Biotech.
I may not recollect.
Okay.
I may not recollect that. Yeah, sorry
Okay.
for that.
No, thank you.
Yeah. I have a question for you. I actually said that there are IL-2 mutants that we are using. So if we have a IL-2 mutant that is having more affinity for alpha rather than the beta and gamma, so what are the applications you can think of? Wonderful, sir. Thank you so much. So autoimmune diseases, because it is going to impact the proliferation of T regulatory cells, and you will have that application for autoimmune disease. Thank you. Thank you, everyone.
Wait, 1 minute.
Yeah.
I think
Yes.
I'm just trying to expand. Use the mic first so that others can.
So do you know how IL-2 is acting on these NK cells, whether the signaling are different from the T cells?
Signaling may not be different. The affinity towards the receptor, there are 3 parts of the receptors, so alpha, beta, gamma. So for NK cells also it is beta and gamma that are more. So that is how it is. So the difference is by the receptor and the molecule interaction. And if you are creating mutants of IL-2 that have more affinities towards a particular receptor, you can use it for different applications. Yeah, signaling remains the same, pretty much same. Yeah.
There's a question at the back. So if someone.
Yes, ma'am. So the PROD IL-2 drug that was conjugated with the receptor, right? So how do you ensure that the IL-2 will dissociate once it crosses the periphery? So it will show its effect. So you said that in the periphery it should not be released. So it is
Right.
engaged with the receptor.
Right, right. So I told you that there is a site where the cleavage will happen, and that cleavage will be done by matrix metalloproteases, MMPs, and these MMPs are enriched in the tumor region. So the tumor cells are secreting MMPs, and that's how it happens.
Yeah. Also, ma'am, second question. The T cells which are destroying the tumor cells. So we know that T regulatory cells are involved in regulation of the cytotoxicity of the T cells. So if alpha chains are more on it, then will it not interfere with the targeted tumor therapy by the T cells? So.
I think it was a bit prolonged question, but what I understand was, if you have more alpha chain, it will be having, a pro Treg effect. So you will have more T regulatory cells. So what was your question? If you have more alpha chain, will it not affect the tumor? Of course, the
Yes,
tumor will be growing heavily because you are suppressing the cytotoxic CD8 T cell response, which is acting specifically against the tumor. That will be suppressed. So you will have tumor growth actually happening in that case.
Okay, ma'am. Yeah, thank you.
One last question. I think there was a question at the back. One last question, and then I think we'll end this session.
Ma'am, you mentioned about there was one mechanism that
Yes.
we can conjugate cytokines with antibody. So can you please elaborate more on that, please?
So I showed you in the prodrug, the cytokine was conjugated with the FC domain, and that is to increase the shelf life, half-life, sorry to use the word shelf life, half-life of your cytokine molecule, because cytokines are smaller molecule, they can be chewed up by proteases, so you need to have larger size so that you can have them in your plasma for a longer time for a better therapeutic effect. Yeah.
And ma'am, there
fusion protein.
Yes.
It's not right way to tell conjugated with antibody.
Okay. 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 Conjugated with FC. Okay.
thank you, ma'am.
All right. Okay. So let us thank Dr. Taruna for a wonderful talk, and thank you.
Thank you, ma'am, for the wonderful talk. I would request Dr. Dipankar Nandi to felicitate Dr. Taruna Madan. I would now request Dr. Sonal Bakshi to felicitate Dr. Dipankar Nandi. Thank you.